Thank you, Cornelius and Emma. That was fantastic, and you guys stayed perfectly on time. So I'm going to move on to our next speaker, and hopefully you can answer a lot of questions in the chat. So next up, we have Marlene Wolfe from Emory University, who's going to be discussing the detection of monkeypox viral DNA in wastewater. And Marlene,
Okay.
I've made you a presenter.
Hello. Okay, let me try one more time to share. Oh, perfect. Okay. There we go. Oh, um, sorry. I did send the presentation in case it's telling me I don't have permission
All right, I will pull that
to
up.
share. I'm so sorry. Let me see if I can.
Um,
Here we go. I think I got it now.
all right, looks good.
Can you all see?
Perfect.
Okay, great. Okay, sorry about that hiccup. Yeah, thank you so much for having me. I'm going to talk about something that's a little bit different, I think, and detection of Mpox viral DNA in a routine wastewater monitoring program. So my name is Marlene. I am an assistant professor at Emory, and I have been for the past 2 years working on monitoring SARS-CoV-2 in wastewater, and now we have extended that to many things, including Mpox. And I'm an environmental microbiologist and engineer. And so, again, a little bit of a different perspective than much of this meeting, but it's wonderful to share these insights together. So I want to share a little bit about wastewater monitoring methods generally. So wastewater serves as a naturally composited biological sample from the community. This is because it includes everything that we put down the toilet, down the shower, anything. So a lot of human excreta, obviously containing viruses and other pathogens, will end up in wastewater naturally composited. And that wastewater contains both liquids, of course, and solids. Our program for wastewater monitoring utilizes the solids because viruses are naturally concentrated in wastewater solids, about few orders of magnitude above the liquid phase. And so we can use that as sort of a already naturally concentrated material to test from. We began this testing with SARS-CoV-2, as many other people have done all around the country and around the world. And what we've seen is a very strong relationship between concentrations in wastewater and cases for SARS-CoV-2. This is just a little schematic here to show you how we kind of can get solids from the wastewater coming into a plant or from different points right after it's settled out. And some of our work showing that there is this sort of enrichment in the solids. So we leveraged that into a high throughput wastewater monitoring program that has been going on in Northern California since November of 2020. We have had between 8 to 12 plants throughout the run of this program in Northern California. We take daily samples of wastewater solids, and then we have a 24 turnaround on testing for results. So the goal is to have this data quickly available for public health consumption and action. And over that time, we've had a bunch of different targets. So as I said, we started with SARS-CoV-2. We've also done a lot of work looking at variants of SARS-CoV-2, seen early introduction of, for example, Omicron in wastewater before we saw it in any cases in a few communities. And then we've also extended into a number of respiratory viruses and now emerging viruses or newly introduced viruses into the population like Mpox. And this QR code here takes you to our research dashboard that has all of our data and also a list of all of our publications. The analytical process is that we take those solids and we just settle those out of the wastewater so that that sample is already concentrated with the targets that we're looking for. We then resuspend it and do a nucleic acid extraction. Our process provides us with both DNA and RNA, and then an inhibitor removal step. Wastewater is a very complex matrix, and there are a lot of inhibitors that make downstream steps, such as PCR and sequencing, challenging. And so a lot of our process is focused on providing a sample that is going to alleviate the effect of that inhibition and allow us to really get sensitive detection of these targets. So we use droplet digital PCR, and we multiplex our targets, whatever pathogens we are looking for, along with several process controls. So as I said, we started with SARS-CoV-2, but all along we have been adding new targets, and this is the general process for that. So first, when we have an emerging pathogen or an emerging SARS-CoV-2 variant identified, we will look to the sequences and we will look to see what's available, what information we have on good targets for that pathogen. We will then either find, sometimes there is an assay in the literature that's available that could be applied to wastewater, and many times we, especially for emerging SARS-CoV-2 variants, would design new primers and probes for mutations and check for specificity. Then we'd procure those primers and probes. As I think probably most of us are well aware, that's the most difficult part of this process, is just getting those in a timely manner to get started, and positive control material, test the set specificity and sensitivity specifically in wastewater, and then start applying that completed assay to wastewater samples that we are regularly receiving as part of our program on a daily basis. So no new samples have to be collected. We're already getting them every day, and they represent up to millions of people, depending on the size of the community. So here's how this process worked for us for Empox. So of course we heard very early on that there was something unusual happening with an Mpox outbreak, and we went to the literature and confirmed that there were many reports of shedding from respiratory secretions, from open sores, and also from urine and feces. And that has continued to be confirmed within this outbreak, that is something that is quite consistently a feature of the virus. And so that made us think this is a good target for wastewater monitoring. These secretions are likely to reliably end up in the wastewater, and so this is worth trying in wastewater monitoring. So we have used the G2RG assay from the CDC assay from Lee et al. 2010. I know that we are currently, as everyone is, thinking about these changes that have been happening in the virus and how that impacts these assays and how it may impact our monitoring program. I'll just say we have had a lot of experience with switching out the specific assays that we're using to contend with that because of our experience with SARS-CoV-2. So I'm not going to focus on that right now, but just so you know, we're well aware of that and are making sure that we're keeping up with those changes. But at the time, we confirmed the choice of these assays by aligning with sequences that were available from this year. Then we ordered the primers and probes and some positive controls, and they arrived within 7 to 10 days, which was actually pretty fast, so that was great. And then we tested to confirm that the assay was working on our platform, that our pre-analytical process provided us with DNA. Since SARS-CoV-2 is an RNA virus that we've mostly been working with. And then we tested against stored wastewater samples and tested for cross-reactivity. And then we began monitoring. We began daily, regular prospective testing of Mpox in wastewater on June 19th of this year in the Bay Area. And then we expanded that into our nationwide program in early July. I'll just quickly show you, we did a comparison of the two different matrices, so the solids and the liquids in wastewater, and we did both the GR2G and GR2WA assays. And what we saw, you can see the green and the pink are the two different assays, and we saw very good agreement between those two in the wastewater. And we saw there were, as had been the case with previous viruses, orders of magnitude higher concentration on a per mass basis of the viral DNA in the solid phase, but that the relationship between those two measurements was significant. So this increased our confidence that we were really seeing Mpox DNA in the wastewater. And so then we have this preprint here. You can see this is on medRxiv. Here's the QR code, and I'll drop some links in the chat. But what we see is that we had 9 sites in the Bay Area and Northern California. We saw the first positives on our second day of monitoring. And what we saw was sporadic early detections, as you can see in this heat map timeline, followed by increasing rates of detection. And then as you can see on the bottom, these are the concentrations per gram in wastewater of the Mpox DNA, increasing rates of detection and increasing concentrations in the wastewater. We partnered with the California Department of Public Health to do an analysis of cases along with the wastewater data. We found there's a very strong association between the Mpox DNA concentrations in wastewater and cases. Cases are on the bottom here. You can see the red is episode date, and then the green is report date. And the Mpox DNA was detected at 5 of the 9 sites prior to or within a day of the first case being identified in the sewer shed area. So we had very good alignment between these results and very good early information about Mpox in the community from these results. This is the just showing you the association between the two, and that we see, we've got on the y-axis the wastewater data and on the x-axis the case data. We're expanding this nationwide. So we have a national expansion of our program, which is called Wastewater Scan. We rolled out Empox testing in early July to all of these sites around the country. We're in the process of scaling up to enroll eventually 300 plants. So we have this network of sites to provide us information on infectious disease trends across the country so that we can say, at a nationwide level, what's going on with the Mpox outbreak, right? So that's really the value as we kind of continue to build this network and show the flexibility to be able to move to a new emerging concern like Mpox. We have had 38 of 54 sites that are enrolled in this program right now have a sample test positive for Mpox DNA in 15 of the 19 states that we monitor. The highest concentrations have been in the Bay Area and Atlanta, and I think it's just a coincidence that's where me and my co-PI, Ali Baim, are, but who knows? And what we have seen is that the concentrations have been leveling out in some of these areas with the highest concentration. So this is in San Francisco, this is in right outside Atlanta. And then, of course, we do see a number of places around the country where we're just starting to see a higher concentrations of Mpox DNA. So that's the value of having a nationwide picture of what's going on through the wastewater. We're currently measuring all of these targets within that wastewater scan program. So we're still doing Sars Cov2. We're still looking at variants. We're also looking at influenza and RSV. We've seen some recent upticks in RSV in the wastewater in the Atlanta area. And then, of course, Empox and our controls. All of our methods and everything are totally publicly available. So I'll drop some links, but everything is free for you to look at. And thank you so much for having us. I know this is a little different for this group. And so I hope that we can have some kind of good follow-up and engagement about this as we get a chance to learn from you in this meeting as well.
Thank you so much, Marlene. That was fantastic. And I think you're going to have a lot of questions. And we actually do have some time for questions if people would like to unmute and ask their questions.
Yeah, I see that there are questions in the chat and I will go through and get to them. I can't multitask that much.
No problem.
I can answer out loud, but I will go through and answer these questions in the chat too.
Would you like to unmute and ask your question? I see your hand is up. If not, I can scroll through and ask the questions in the chat. What did you use to extract nucleic acids from Fabricio Colosimo?
Yeah, so we use the Comagic 360. It is it's a total nucleic acid extraction kit. And I will drop a few links here because I'm having a brain break on what the exact name of the kit is, but I have it available here.
Okay, and let's see, there is another question. Can you include the URL to your protocols page in the chat, please? And I think there's a couple of things that's already linked. Am I missing anything else? I think that is it. All right.
I think this is a good, if I can answer one question out loud from the chat. Is that okay? This question from John, what is your strategy for working with health departments and places where we're measuring Mpox? That's extremely important. And from the very beginning, this has been a close partnership between us as academics and researchers and utilities and local public health. And so we've developed over the course of 2 years of monitoring Sars Cov2 really close relationships with these public health departments in Northern California and the California Department of Public Health, who are collaborators on this paper looking at Mpox. And so we've had a lot of support from them all along and a lot of encouragement from them to do this monitoring. As we've extended into places around the country, our goal is never to surprise anyone with information from their wastewater about Mpox. And so we reach out to the public health departments, we work with the utilities to make sure that they know that we're measuring for it and that they get the results right away and that we're available to help them interpret those results. The interpretation is if we see Mpox DNA in the wastewater, we know that there's at least one person in that community who's infected and shedding the virus. This preprint allows us to give a little more information around that relationship and the trends that we see, and helping us understand that as we see increasing and decreasing trends of the viral DNA in wastewater, that is very likely telling us something about the trends in the cases in the community. And so our goal is to never surprise anyone, make sure all of our information is openly available, and to be able to provide support to communities for understanding this data and integrating it with the other information that public health departments will be using to make decisions about public health action, which is not just this data, but we've found that we've had a very positive response. People find this data to be very supportive to their outbreak response.